Good evening. This is the final journal club of 2021-22. Yohann is presenting an actually good paper called Cancer Cells Use Self-Inflicted DNA Breaks to Evade Growth Limits Imposed by Genotoxic Stress, published in a little-known journal called Science in April, so very recently. And go.
All right, so I just want to apologize beforehand for the delay. That's my fault. But without further ado, let's get into this paper. Let me just share my screen real quick. 1 second. All right, cool. So, all right. So let me. All right, let me go back real quick. All right, so like Dr. Char said, the paper is called Cancer Cells Use Self-Inflicted DNA Breaks to Evade Growth Limits Imposed by Genotoxic Stress. And basically, the premise of this study is it highlights a survival adaptation that cancer cells have that cancer cells use to fight against or hinder genotoxic stress, which is irradiation. So basically, imagine a cancer cell that's being shot by radiation. We're talking like cancer patients and such. So that cancer cell contains a nuclease called CAD, which I'm pretty sure stands for I have it right here. Yeah, it stands for caspase activated DNase. So what CAD does, when CAD is present inside a cancer cell, it essentially it promotes DNA breaks within the cell, which then keeps the cell at a G2 checkpoint, which means that it prevents the cell from creating malformed DNA, to put it in that. I could put it that way, I guess, because that's like a survival adaptation. Because if you're going to create deformed DNA, the cell's not going to be viable anymore. So in order for the cell to protect itself, the CAD is used to increase the number of DNA breaks within the cell, which then produces a G2 checkpoint, which then prevents the cell from creating any more cells. So then after the irradiation, it can then keep producing cells that aren't going to be affected by the radiation. So here in the diagram is showing two different cells, a cell with CAD and a cell without CAD. So with the cell with CAD, you can see, like I just said, there's no damage, and then there's the radiation, then there are the breaks, and then the repairs. So then CAD creates the DNA breaks, and then which leads to G2 checkpoint proficiency, and then the CAD deficient cell or the CAD knockout cell. There are breaks, but then there aren't enough breaks, and then the cell just keeps producing DNA, which isn't viable, which then leads to cell death. So that's basically what the paper is talking about. Now, in this first figure, wait, Dr. Char, do you have anything to say, or was that?
That was pretty good.
That was pretty good.
Yeah.
All right. Sorry. Oh, why do I keep doing that? How do I go back? All right. Sorry. All right. So in this first figure, 1 second. All right. So in this first figure, the researchers measured the amount of DNA breaks in different types of cells using the alkaline comet test, alkaline comet assay, which is basically like a way to measure the amount of DNA breaks within a cell when it's being irradiated. And in the diagram, if you look closely, you'll see this measurement, it's like IR, I assume that means irradiation. And then it says right next to it 8GY, which is basically a measure of radiation. And it's 8 grays, and I'm pretty sure there are 800 rays in 1 gray. So in total, it's 800 grays, but that doesn't really matter. It's just the amount of radiation that's being like shot into the cancer cell, or like in this specific in these specific figures, it starts off with the cancer cells, and then it starts out with the and then it ends with like a multitude of cells, like in figure D. Like there are cancerous cells, and then after that there are just regular human extracted or regular cells from, I think, animals. But let's tackle figure A real quick. So figure A is basically showing a wild type cell from, I'm not sure where the cell comes from, but I just know that it's a cell that I'm pretty sure is cancerous. Yeah, it's a cancerous cell. And it shows it with it shows the cell with CAD and without CAD. So the alkaline tail moment measures like how many lesions there are. So in this, and then there are also intervals to this. So like there are 10 minutes, 2 hours, 6 hours, 24 hours. So then it just shows like how many lesions are made during those intervals, if that makes sense. That's pretty much it. And then actually, yeah, that's pretty much it.
Yeah, so these HCT 116 cells are actually from a male patient who had colon cancer. It's an immortalized cell line that lives, forever. They're used as cell lines.
Okay, yeah, I was a bit confused on that, but yeah, I wanted to. I think I have it right here. So yeah, so figure D, I just want to talk about that a little bit more. So in figure D, there are 4 cancer cell lines and 2 non-cancer cell lines. So in the non-cancer cell lines, you can see that the last ones display the least amount of DNA breaks. I'm assuming that that's because that shows that that's like a feature that only occurs in cancer cells, because the cancer cells they produce more lesions when shot with the irradiation, but the non-cancerous cells don't even produce a close amount to that of the cancer cells in number of lesions. Correct me if I'm wrong.
No, I think that sounds right. The healthy cells can actually tolerate quite a bit of irradiation.
Right.
Whereas cancer cells, if irradiated, either die, which is why we use radiation therapy. They either die, go into premature apoptosis, or they go into, or it makes them worse. And that's what I think this paper is trying to address: is how do those cancer cells that become worse, how do they become worse? Fair?
Yeah. Okay. All right. So that's pretty much it for this figure. So next figure. All right. So this figure, so part A in this figure shows immunoblotting, which is similar or is what we did in the summer lab intensive. I mean, sorry. No, it's not.
No, so the gel is run the same way, but it's not quite the same thing. This is when we did it last year in the summer lab intensive, we learned PCR and we ran DNA on a gel. This is protein isolation, and the proteins are run through the gel. This is an immunoblot.
So is that
Western blot?
Is that the chromatin? See, I'm a little confused. Is that the chromatin or is that
DNA? So it's a chromatin fraction. It's It is the proteins that are isolated from the DNA. When you break open a cell, you can actually do that just by using certain types of salt concentrations and chemicals. You can keep the DNA and its associated proteins. What proteins are associated with DNA? Chromatin, right?
Yeah,
So then
well,
what you do is then you need to isolate, well, which chromatins are they? Because there's multiple chromatins. So which histones are they? And so what it does is, like, if you look at the bottom one, H3 is histone 3. Now, it doesn't matter what type of cell it is. Histone 3 is a basic important piece of chromatin. So you should have the same amount of histone 3 in all samples. What makes this different from what we did is that once you run the gel, then you actually put some sort of antibody on top of it, which will then make whatever it's usually done with radioactive stuff. And it'll attach to the protein for which it has the antibody binds. So the antigen it binds to. So like those antigens will be P53 or ICAD-L or CAD. So it'll tell us which proteins are there because it binds to them.
See, another thing that I found kind of interesting is I was wondering why CAD showed like the most amount of protein accumulation at the 24-hour mark, because in the other like P53 and ICAD it's less, because I know ICAD is the inhibitor of CAD, but I don't know if like they work like together.
Well, if this is a timed assay, you would 1 hour. The fact that there's anything in 1 hour is kind of remarkable, because it usually takes something like on average about 4 hours, for 3 to 4 hours for genes to be turned on at all. So that's why you see a big thick line at 3 hours, but not at 1. So what this is doing is it's trying, you are irradiating them. Yeah, that's what it is. You're irradiating them and then timing how long it takes before CAD is turned on. And in response to the CAD, you need something to keep it in homeostasis so ICAD gets upregulated, and to see if P53 is actually also up. That should not change how much H3 there is.
So like this is what we were talking about earlier. So H3 is the active control, or what do you say?
It's the control. It's called a loading control.
Loading control.
Yeah.
Right.
It's you want to make sure that you put the same amount into each well. Now, if you look at it, you can see that that H3 is darker in that 6 hour band and maybe in that NT band. And what you do in order to figure out, well, how much protein we're talking about is something called densitometry, where you find out the density of blackness on that gel relative to the control. So you actually do it as a fraction. So CAD, the amount of CAD at 3 hours divided by the amount of H3 at 3 hours. The amount of CAD at 6 hours divided by the amount of H3 at 6 hours. It's a pretty simple classical way of measuring whether a protein is being made or not, which it clearly is. I mean, you can see a difference over time. So it clearly is.
All right. Yeah. So moving on to figure B. Part 2, figure B. So here there's a protein called M cherry that shines red under a mic. Is this under a microscope, or is this like so it shines red under a microscope, but it only shines red. What do you say?
I think it's a confocal laser scanning microscope like I used.
Right. But it only shines red in this scenario when there are DNA breaks present within the protein, within the cell, and then those proteins shine due to the DNA breaks.
let me say a couple things before you continue that you may not know. Tunnel is a chemical method we use to see if apoptosis is active, if apoptosis is turned on. Okay. If you see any fluorescence, that means that the tunnel stain is positive, and whatever cell you're looking at is going through apoptosis. So the one on the left, it does not. The one on the right is. Okay. Then down in the bottom row, you see it's the DAPI. DAPI, that's an acronym for a really long name. I can't remember off the top of my head, but what it does is it stains. That works like a stain. It stains for DNA. The chemical actually inserts itself in the minor groove of DNA. And if you shine a UV light on it, it fluoresces this beautiful blue color, which is what the blue you're seeing. The M cherry is connected to the LAC, which is on a promoter for whether or not you are activating tunnel or whatever, or not tunnel, apoptosis.
Which well, the DNA breaks do they translate to apoptosis? Like do they like do they mean apoptosis, if that makes sense? Like do they mean that apoptosis is happening?
If you're seeing tunnel, it does, yes.
All right.
But WTI CAD, I don't know what that is. Well, I've been looking for that. What is that?
WTI CAD.
You know,
yeah, I have no idea. I mean,
WT usually means wild type. Wild type ICAD.
Oh, I think that's the
thing
HCC
that responds.
cells.
Right. The thing that responds to breaks, right?
Right.
It responds to CAD breaks. Gotcha. Okay. All right.
Moving on.
Yep.
Anybody want to do this figure?
You know, you don't have F, right? 2F?
No, I don't. I don't have 2F, no.
Because 2F has this really, really beautiful drawing.
You wanna take over?
Yeah, let me just show this real quick. For those of you, so pin me for a sec, people, so you can see this. Stop. Just happened. That's not what. There it is. Okay. It has this really beautiful figure here, kind of showing this thing here. Can you see you can see my mouse. This thing is a histone. And histones are made up of 8 proteins in quaternary structure. So you know how you have primary sequence, secondary sequence, tertiary structure. And then so like secondary is alpha helices and beta sheets, tertiary is how the whole thing folds, but then quaternary is how the whole thing folds and then interacts with another protein. It takes about 8 of them to make one histone that's used in a nucleosome. And you can see also that the DNA, which is this darker band, wraps around the histone 2.5 times. And then they draw like this. And there's another type of histone called a linker histone in between the pearls on a string, as it's called. The model is called pearls, which are the histones on a string, which is the DNA. On each side of the linker histone is the CAD itself, which does the DNA breaks.
So that's the Pac-Man, if you will.
Yeah. And the Pac-Man is the thing doing the breaks to try to isolate the damage to the DNA.
All right.
Okay. All right. So now back to you.
All right.
What figure was it? 3?
Yeah, we're on 3.
Okay, so no takers. Nobody wants to try. All right, I'll do it. Let me see which ones you actually put on there. You put the top stuff, A and B.
Yeah.
Okay.
Those really summarize like the whole fig. In my opinion, the whole figure.
Okay. So, well, hold on. Good. Okay. So check this out. All right. So you can see on my screen, I just did a quick Google search for ATM ATR because I wanted to show you the intense signaling pathway. Okay. So when you have DNA damage, I think this is a good example. Let's see if I can make it big. Yeah. When you have DNA damage, that DNA damage activates ATM, which is a protein, which itself associates with and activates ATR, which then has this huge cascading effect. In basic honors biology and AP biology, when we think about proteins having effect, we think of one protein having an effect on one other protein, which has an effect on one other protein, like a game of telephone. That's not actually how this works at all. It's like one protein can probably bind to and activate 15, 20 proteins. And each one of those can activate 15 or 20 proteins. So it's way more complicated than we let on when we're teaching this at the basic level. ATM is a classic example of this, because I remember when I was in graduate school, we read a paper which made it into science, and Dr. McMahon and I didn't think it deserved to be in science at all, because it pretty much told us that ATM activates a lot of stuff. We already knew that. Like, that's not interesting. But one of the things that it activated was a circadian gene, which they have not actually included in this. But you can see, let's look at ones you might care about. So in this drawing, you have P53. That should look familiar, because that's what you see in honors biology when we talk about cancer. Here's another one, BRCA, the breast cancer gene 2 and the breast cancer gene 1. Both of them are turned on by ATR. P53 again. GAD. Let's see, other ones I care about. Junk's a fun one. Junk kinase. So you have this huge cascade of activation that, I don't know if you noticed this, but on the outside of this figure, it actually shows you what part of the cell cycle it affects. And a bunch of these affect DNA repair. Obviously, if you have DNA damage, you're going to get DNA repair. So back to the figure. All right, so go back to Johann's presented screen. And on the left column is the control. The right column is irradiated, but they have actually introduced I did something like this in my research. I think I've shown you guys that I had a mouse that its brain would glow during the daytime, and then it would stop at nighttime, and then it would glow during the daytime, and then it would stop at nighttime. That was a protein called green fluorescent protein. That's what GFP is here in this figure. Green fluorescent protein, and it is attached to the promoter for CAD. Okay, so if CAD is activated or downregulated, you will see a change in the GFP. You can see that between the left and the right one. So the irradiated one is actually decreasing. M cherry is a red variation of green fluorescent protein, which is really bright. Red is actually kind of hard to see under a microscope. This particular one is useful because it's super bright and easy to see. And so this will show whether ICAD is also then changed. And what you'll see is if you measure the raw fluorescence, just make sure that that's what RFI means, because that's what I would call it. Sure, I guess that's what it means. They don't actually say.
So the green the M cherry is detecting the ICAD, not the cat.
Correct.
Right, cool.
Okay. And you'll see that in the graph, the lines are pretty much even. You don't see any significant differences between those lines.
Mm.
Okay, so that's in a normal irradiated situation. Okay.
Yep.
All right. That's what you would expect. There's a major problem with this, but they actually do a pretty good control to control for it. But and I'll just tell you what the major problem is. This is something I would love to teach you guys how to do, but they're using a microscope that probably costs about $250,000. And when you are firing a laser at a cell and you're seeing that the fluorescence dims like you do on the microirradiated column, it could simply be that you've cooked the tissue. And so the reason it's not fluorescing is because you overheated it. You cooked it with a laser. And this is something that it's funny. People who are new to it don't realize that. And then they're like, oh my God, I got to change. And it's like, no, you just killed it. You just lit it on fire, essentially. But they actually have a control for that in the middle band. If you see, there's kind of some banding right in the middle. They actually did that on purpose. Okay, now B is making the same measurements, and they are treating the cells with a chemical called dimethyl sulfoxide, DMSO, or a chemical DMSO with an inhibitor for ATR in it. So both of them have DMSO. So when they do this, when they introduce you'll see the red dots. The red dots have the inhibitor for ATR. You actually see that there is a significant decrease in that reflectivity of M cherry because the ATR is the direct contact for the ICAD. So if you turn it off, it can't actually behave the way it's supposed to. All this is showing, and I don't know. I actually don't know why they. Oh, I do know why. Never mind. So in B, the one on the left is for CAD. It's an inhibitor for CAD. The other one is ICAD. Both of them you see are depressed. The lines are depressed, which means that it is ATR that is the pathway through which CAD is working. I think that's the simplest way to say it. So you freshmen, the 6 of you who are here or whatever, because there's only 9 people in the talk now and 2 of them are teachers. Notice that the figure 3 figure legend starts with what the actual result is, that ATM-ATR signaling to ICAD is the thing that's regulating CAD-induced DNA breaks.
So that's basically the summary of the figure.
Right. And that's kind of why even though I took a while to walk it through, what's the conclusion? Well, exactly what it says, that
right.
when you actually activate ICAD or CAD, it is working through a pathway called ATR. Why is that significant, Yohann?
Why is it significant to know that it's working through that pathway? Because then we could target it.
Exactly.
And that's one of the conclusions. But I didn't specifically say that. I said that it happens at the G2 phase. G2 phase, like of mitosis, I think.
Yeah. Okay. G2 phase of the cell cycle.
Yeah, cell cycle.
G2 is different from mitosis, right? Okay, next.
All right. Figure 4. Anybody? Come on, Miss Liff.
She's cooking.
She's not
she's
there.
Cooking. Don't make her do that. She's there, but she's cooking.
Nick,
Tyler, why don't you give it a go? You want to try?
All right.
Okay. I'll help you out.
I read the stuff at the bottom.
Yeah, you chose. So you only want to do E through G, Johann?
Yeah, E through G.
Okay. Oh, because it summarizes the ones above. Yeah, that's
Right.
smart. Yeah.
E is the relative colony outgrowth of CAD depleted cells after IR exposures. F is do you want me to read the whole thing?
Don't read it.
Well,
what is it of? What is it actually showing us?
like, what is E showing us?
Well, let me ask. Oh, never mind. It's right there. So we're still looking at the same cells. We're looking at colon cancer cells from some guy, right? Those HCT 116 cells. So that's what they're calling a colony. Okay. So what's the blue, Tyler? What's the blue line showing us in E?
Sure, I'll be right back.
Okay. Well, what's it say? What's the label say, Tyler?
SIC.
Okay. So did we talk about in honors bio, did we talk about small interfering RNA? I think we did like super briefly in like chapter 8 or 11. Does that sound familiar, Lila?
Yeah, I think it might have had to have been like pretty brief because I don't.
Oh, you're
remember.
much clearer now.
Oh, yay. Great. Okay.
Okay. So small interfering RNA is used to interfere with translation, to essentially get in the way of the ribosome so that it can't do anything. So if I use a small interfering RNA for CAD, then what's the blue line mean, Tyler?
Would it be the growth of the colon cancer cells when they're interfered?
Right. And so what are you seeing though? Is there a difference?
The decrease?
Yes. Significant. Notice the star, right? It is a significant decrease from the UNC, which is a scrambled type. It's that's just a control, which we can explain later, but that's just the control. So then what's F? This one's the most interesting one is F, I think. Do you know what KO means?
No, I'm gonna guess it means
Do you follow boxing?
knockout.
Yep. That's what it means. Yeah. So CAD knockout means that they have gone into the cells and removed the gene for CAD. They've knocked it out. And doesn't it look really similar to the one on the left to E?
Yeah.
So that's just more. That is more evidence that CAD is the thing allowing for repair even in cancer cells. Because remember, the point here is that you get cancer. One of the treatments we do is chemo, which is a type of directed mutagenesis. The other type is radiation. We irradiate you to get rid of the cancer. But they're usually irradiated cancer cells die, but sometimes they don't. And that's like radiation, I'll say defiant or resistant cancer is really hard to treat. And those people usually die. But what if in this paper, they've actually identified how to get around that? Like, what if you go, like you do a round of radiation, and then we test you again, and the cancer's gotten worse. We go, oh, you're one of those people. Hold on. We're going to give you small interfering CAD RNA to inject it directly into the tumor so that it can't repair itself after we irradiate. Then they irradiate again, cancer goes away. See how cool that is?
Mm-hmm.
Like one of the problems with the 2020 AP exam was the free response question asked, how do you cure cancer, which you can't do. However, studies like this get us one step closer to the College Board, maybe being right in 20 years, but certainly never right enough to give that question to seniors and high school students in general, right?
Yeah.
So that was not fair, and they should be ashamed of themselves. So G. How do they even measure it? See, I don't know how they measure genomic instability. Like, how do you even do that? Oh, micronuclei. Oh, where the nuclei are actually degrading, they're degenerating, and so you could see that in the knockouts, unable to respond, unable to fix the damage, they are dying at a higher rate by day 3. It's a pretty good turnaround. Okay.
So does the NT means like start or like
NT?
Yeah, like from like the time intervals.
That's the NT is the start, right? They haven't done the procedure yet. Yeah.
Right. Yeah.
This is cool. This is the best paper we've seen.
I have a question. Why is it fixed at 8 grays? Does that
that's that I don't know anything about the way that radiation is graded, or the types of radiation. I've never done anything like that. So I would think the higher the number, the greater the radiation risk. We were talking about this today in my AP bio class, where what's the number one, what target is number one on every terrorist's list for a nuclear weapon, right? It's New York City.
Mm-hmm.
And so we were discussing what would happen if a nuke went off in New York. Would it affect us here? And the one issue that we would have is that radioactive radium and iodine would probably float on the wind, but usually the wind goes west to east, not east to west. So there might be a little circulation, but it will mostly go towards Connecticut. And the way that hurts people is that your thyroid takes that and takes the iodine, radioactive iodine, and replaces its iodine with the radioactive type, which then causes cell damage and death. So what you want to do is you want to take an iodine pill just when this happens to kind of avoid that. Now, it doesn't stop other types of radiation, but if you then increase the amounts, and I think that's what those levels mean, like the increasing amount of radiation that you're exposed to. But I don't know. Miss Liv, you don't know anything about that, do you? Is she still here?
So the worst problem would be the amount of groundwater contamination.
Oh.
Not even wind. Because in any situation like that, it's going to rain because you're disrupting cloud surface cover. And then that rain is going to run off into your groundwater.
So non-immortal people will probably die.
Yeah.
So I'm fine, but everyone else is dead. Well, that sucks for you people. But okay. Keep going, Yohann. Let's finish up.
All right. That was it. All right. So yeah. This study basically, like I said, can help lead to research that essentially targets the cancer cells at their G2 cycle checkpoint, so they can more efficiently eliminate the tumors targeted at the checkpoint, so they don't get to that checkpoint I don't know how to say this. Like, they don't get to that checkpoint stage where they prevent themselves from creating
cell death.
Like, well, cells that aren't viable, because if they do, then that ultimately leads to cell death.
Okay.
So if they do that, then the cells would then create those cells that aren't going to function properly, which are then going to affect other cells around them, and then they'll just kill each other. And lastly, so it just contributes to a possible cure to cancer, as like we stated before.
Okay.
Yep.
All right. Questions? I'm going to ask a question I haven't asked in a while. Because the last several papers were either in journals that I wasn't really familiar with, or I didn't understand them enough. But does this paper belong in a journal of as high a caliber as Science?
Definitely.
Yeah, I think so too. Yeah. This is one of the coolest papers I've seen in a while. Yeah. Questions, comments? Anybody else? Here at the end, 13 people are now on instead of 9 like they were 10 minutes ago. So I don't know who just signed on so that they could be like, yeah, I was here the whole time. But it was probably Sam.
I've
Miss
been
Liz,
here the entire
She
time.
has been here. She has been here.
Sam bagged me, so she's on my my list right now. She bagged me for kittens. Poor kittens was looking forward to a nice vacation.
I wanted to take care
With
of
the
her.
Cepheus. All right. Any last notes, questions?
No, not really.
Okay, so planning for summer. I'm ordering as soon as golf is done, I'll finish ordering stuff for the summer. I'll make sure that we have that we're going to the Met. We're not going to be able to use coach buses because there's just too many people, I think. I may even try to go and get my license maybe this weekend. And I am keeping my eye on the weather. If a clear night shows up, we may shotgun call something. It won't be mandatory. It'll be for fun to bring your telescopes to the what is that? That central area called oh my God, what's it called? The courtyard. Jeez.
Wait,
to the courtyard. Yeah.
Dr. Char, I have this tiny telescope. It's I definitely didn't like. I'm definitely not gonna steal it from my brother, but it's like this tiny telescope, right? I don't know.
Yeah.
It works pretty well. Actually, let me go get it real quick. Come on.
Yeah, I mean, all of you should bring your telescopes no matter what, and cameras if you have them, and we'll train them at the stars if the stars will ever show up. But it does not look good. Hold on. If I go.
What are you making?
I don't.
Just decorated sugar cookies.
Yum.
Does this work? This is actually.
I think it's missing an eyepiece.
No, yeah. The eyepiece is over there. I have
Okay.
it.
Well, it's a refractor. It's an inexpensive refractor.
Oh yeah.
It'll work a
definitely.
little bit on something large, but it's not going to be as sharp as some of the as Dr. Kerr's, for example. Dr. Kerr has a sweet telescope. All right, so it looks like, as you can see, look at this. Here's the nighttime, right? Red. Maybe on the Sunday the 29th, but I'm not here. So I don't know. We'll keep an eye on it, and if I am in town, even over the summer, I'll call something together. Like that week we have off, maybe I'll do that. We'll see. Okay. You have a cat. I forgot about that. Does the cat want to be part of the summer AMI program?
That's such a terrible thing to say. You want
I was
kill
trying
my
to be
cat?
inclusive. I was just trying to be inclusive.
Now my dog is barking.
What if the cat wants to be involved?
How would it be involved?
He wants to kill my cat.
Would we do a dissection of it or something?
I was thinking that'd be fun. At least the cat would then contribute to society in some way
That is
possible.
so mean.
It's for science.
It's for
Now you're all cat haters.
mic drop. It's for science. All right. I'm gonna go eat. It's been wonderful, and so I think that was a pretty good year. We're gonna do all our capstone stuff and finish up, and it's gonna be great. Tomorrow is the last day for the seniors and the NJAC championship. I'll be there in the morning, and then I'm not there, and then I'm not there probably at all on Wednesday. So I'll be back. I'll try to get there Wednesday afternoon if we can get done in time, but Thursday I'll be back, and then we're gone again. So.
Where can we find the recordings of these?
There, I haven't, the last two I haven't done yet, or this one. So I've got 3 that I need to encode and post. They're on our YouTube channel, and they're also on the website. If you go to the website and go to Journal Club, you'll actually see they're posted all in there under each person. Okay. Yeah. So I'm going to actually I'll do these videos tonight, and then we'll be done for the year with AMI, waiting for the summer thing. Hopefully you all have your AMI notebooks. Good job, Yohann. That was good paper. Yeah. All right. Good job, Miss Liv, with whatever it is you're making. Okay. See you guys later.
All right, see you, Dr. Char. Thank you.
End 2022.
Thank you.
Yes.
Signing off.
Bye.
From AMI. Goodbye.
Sorry. Quick question. Can you hear me?
Yeah.
Yes.